The molecular lab bench allows you to amplify DNA using the polymerase chain reaction, PCR technique, run an electrophoresis gel, and determine the nucleotide sequences of amplified genes. The equipment used in this lab also includes the Eppendorf tubes and tube rack, reagents and primers, and the pipette. To choose a DNA sample, simply select a species from the species selector. The PCR machine is used to run the polymerase chain reaction in order to amplify the DNA sample. After running the PCR, you can run a gel electrophoresis experiment to determine the mass of DNA mixtures, or the sequencer to determine nucleotide sequences. The laboratory clock can be used to accelerate the experiments, and the cost calculator keeps track of the costs associated with the equipment you use. Clicking the red bin will reset the lab to its initial state. The amino acid and nucleotide charts can be referred to during the experiment. They can be enlarged, sized, and moved for reference while performing the experiments. Finally, you can record your observations in the lab book, use the clipboard to perform pre-configured experiments for this lab, click the Help icon for detailed help on the labs, or click the Exit icon to leave the molecular lab. This concludes the overview of the molecular lab bench.